In this video, we look at key area 5, which covers the circulatory system. We're just going to start with some recap questions. And the first two, you'll notice I've picked sex-linked inheritance for all three of these questions, just because it's more tricky and I'm a sadist. And the third one, I've decided to give you someone with Klinefelter's as the male parent. There's a variety of genetic reasons this is not really sensible, that doesn't matter. Have a go at the three. It's a very good opportunity to practice your Punnett squares, which is a hint. So a genotype and phenotype of the offsprings. Okay, the formatting on this is not amazing, but for the first parental cross, we have the male, so X with the recessive allele, and Y with the masculine. And we have the female who is X, and then she's a heterozygote, or in this case, because it's sex-linked, we can call her a carrier. So we put the male possible gametes down here. You could be X with the recessive H or Y, so we can add those in. So X with the recessive H would go in this one and this one. Y would go here and here. And the female genotype is X with the dominant allele, X with the recessive allele. So if these two cross, you would have X dominant, X recessive. So you would have a female phenotype who was a carrier. So phenotypically, she would have whatever phenotype the capital H gives. So if it was sex-linked recessive, then she would not have the condition. If this was sex-linked dominant, which isn't really something we cover at Higher Human, then she would have the condition. In this case, you have a female who has the recessive phenotype, whatever that is. So in here, in this case, we have an X with capital H and a Y. So this is a male with the dominant phenotype and a male with the recessive. So you have a male with the dominant and a male with the recessive phenotype. You have a female carrier and a female who is unaffected here. For the next one, we have, again, a male and a female crossing. This time, instead of having recessive male, we have a dominant male, and in the female genotype, we have homozygous recessive. So if we do the cross again, puts the male X and the Y down here, and the female X and the X here. In both cases, the female is going to give her X with a recessive allele, so actually her genotype is, in terms of Punnett square, irrelevant. 50% of the time, you will get the male's X combined with an X recessive, and 50% of the time you will get a male produced who will be X recessive. So that's what we see here. So again, there's two phenotypically, basically the females are going to be phenotypically capital H, and the males are going to be phenotypically recessive. And in the final one, I've given you some with Klinefelter, so XXY. Quite often, as I said, people with Klinefelter's, there's things called mosaicism, but it doesn't really matter. Quite often they would be infertile, so this cross wouldn't necessarily produce offspring, but if it did, this is how you would do it. You just add an extra square. This is never going to happen in your exam, especially this year. As a person who writes your exam, we haven't gone off-piste and put something in that's not in the course. This is just proof of concept, allowing you to try out a different type of Punnett square. So we have a heterozygote or a carrier in this condition. No, we don't. So this Punnett square is wrong. Anyway, so we have the homozygous recessive, so X with a small h, X with a small h. If that is crossed with the male's X recessive, you will have X with a small h, X with a small h, X with a small h, X with a small h. In this case, you will have X with a big H combined with X with a small h. Because the actual genotype you were given here is homozygous recessive, you can kind of strip this down to a single column because both of the two columns should be identical to the genotype you were given here. So that should be a homozygous recessive female, so X small h, X small h. That should be a heterozygote or a carrier phenotype genotype, so X with a big H, X with a small h, and that should be X with a small h and Y. So, just getting on to a little bit of the actual course content. So why do we vasodilate and vasoconstrict? This from the starter task I gave you and also from National 5, you might know the answers. So vasodilation, we will cover in the next video the difference between vasoconstriction and dilation, but to simplify it, if the blood vessels relaxed or smooth muscles, you get blood flow to the extremities and to the skin, which makes you quite flush, and it's good at getting rid of excess heat. If it's quite cold, your smooth muscles in the arterioles will contract, and it will prevent blood going to the surface of the skin and to the extremities, and you'll vasoconstrict. So we're going to learn a little bit about the structure and function of the blood vessels. So you could try and list as many things about the circulatory system you know from National 5 or general knowledge. So what is the cardiovascular system? The cardiovascular system is in the name. Cardio is the heart and vascular is the blood vessels. So the cardiovascular system, CVS, moves blood around the body. This is important for heat. So your body spreads heat through the blood. It spreads nutrition out to, again, the central organs and the extremities. It removes waste products through the blood, and it transports hormones. So in National 5, we've talked about hormonal transport and how it works, and we've looked at menstrual cycle hormones and the male hormones. We'll go on to look at things like insulin later on in the course. Two become one. I like to think of the heart as having the right pump and the left pump, and they just so happen to be attached together. So the right pump takes in deoxygenated blood and pumps it to the lungs. And the left pump takes oxygenated blood and pumps it out to the body. It just so happens that the left and the right side are in the same organ. If you just remember completely separate pumps, it sometimes helps to remember what the right side and the left side do. So we have three types of blood vessel that we're going to quickly go through today. We have the arteries, we have the veins, and we have the capillaries. So arteries and arterioles, which isn't really a term we cover a lot in the course. Arteries go away, so A and A, arteries away from the heart. The heart contracts in the ventricles, so the lower chambers, and it pushes blood out at high pressure. So this means that the pressure in the circulatory system is highest when the blood leaves the heart, and we say it decreases as it gets further and further away from the heart. It's something called turbulent flow. So as the vessels get smaller and smaller, the pressure actually decreases a bit. Arteries have quite thick walls, so they have an outer layer of connective tissue. So this is like the outer, if you think of a wire, it's got the outer flex, and then it's got the inside bit. So in an artery, the very outermost bit, the covering, has elastic fibers. In the middle of the wall, it has smooth muscles with a little bit more elastic fiber. And then on the inside, it has what's called the endothelium. So that's the layer that would be in contact with the blood. The elastic walls of the arteries will stretch and recoil to accommodate the surge of blood after each contraction. So if it was a hard, like stony structure, every time the heart pushed, it would strain the walls of the arteries and they would eventually break. So they need a little bit of elasticity so they can withstand that pressure wave and then carry it on as the blood moves through the arteries. So leaving the heart, we have the aorta and we have the pulmonary artery here, sure. The three types of blood vessels, capillaries and next, they are allowing gas exchange and fluid and nutrient exchange in the tissues. They have very thin walls, so their walls are single-celled. I'll show you a picture of the three blood vessels, and you'll see that capillaries are kind of like the most basic level. If you imagine just a ring of single cells making a circle, that is your capillary. So they have an inside and an outside, but it's only separated by a single lining of cells, what's called the endothelium. And then on the inside, we've got the blood, and on the outside, we've got the tissue fluids. So the capillary net goes through all of your organs and all your tissues, and it allows the transfer of things in and out. There's three types of blood vessels. The final one is veins, and again, venules we don't talk a lot about, but we do a little. An outer layer of connective tissue containing elastic fibers. So this again should remind you of the arteries. They have the same overall structure as the arteries, except for valves, but their inner muscular wall is a lot thinner. So that means that they actually have a larger lumen. So if you had the same sized vessel and it was a vein or it was an artery, they could be the same size, but the artery would take up a lot more of the internal space by having a thick muscular wall. The veins don't have that, so they tend to have a wider internal hole, which is called the lumen. Inside that lumen, they have valves, so that means that as the blood moves forwards, when you then stop contracting your muscles, if the blood tries to flow back the way it's just come, it can't, because valves close behind it and stop that happening. So this is the picture I was telling you about. So we can see arteries. They have a layer on the very outside, which is smooth and contains elastic fibers. We'll then have the muscular layer on the middle, and the very inside layer is just single cells of endothelial lining. In the vein, we have the same basic system. We have the outside, which is connective tissue containing elastic fibers. You can see this inner muscular wall is a lot thinner, and then we have the endothelial lining. So the same sized vessel will have a bigger lumen in the vein because it doesn't need to have as much muscle on the wall. And at the capillary level, these are single cell thick, so it's just this very inside layer of the artery and vein. So that means that nutrients, gases, things from the inside can easily diffuse out, and waste products can easily diffuse in. The holes are big enough to allow that to happen, but as we talk about later, when we look at pressure filtration, the holes are not big enough to allow proteins to get out from the plasma into the tissue fluid. So on page 159 of the Higher Human textbook, you would carry out an activity to demonstrate the presence of valves in veins. If you're someone who has a lot of the little blue veins running around the body, you might have done this before. You can kind of do it on your wrist. It's easier to do on your, I think what's called your ACF, your antecubital fossa. So if you have a nice big bouncy vein on the inside of your elbow, you can press a finger just above the vein, and then you put a finger just below that, and while keeping the finger at the top of your arm in place, you can push with the other finger down the vein, and what you see is it disappears. If you remove that bottom finger and keep the top one nice and pressure, the vein should not appear again. So what happens is when you apply pressure at the top of the vein, you're cutting off the blood supply. When you push the blood away using your bottom finger, the blood is pushed past a series of valves. And what that means is when you move your bottom finger out of the way, the blood might try and rush back to where your top finger is, but it actually can't because there's valves in the way. And then if you remove your top finger and squish your hand a bunch, you should see the blood comes back. So with veins, you can push the blood past a valve, and if you've got something stopping blood filling from above, basically your veins completely close. In the next section, we'll look at vasoconstriction and vasodilation. So hopefully that made a bit of sense, and you are able to understand the concepts. Just quickly, I'm going to demonstrate this. So we have looked at blood circulating from the heart through the arteries, through the capillaries, and then to the veins and back to the heart. We talked about a decrease in blood pressure as the blood moved away from the heart. We then looked at the structure of the arteries, capillaries, and veins. We talked about the endothelium, which is this inner central lining around the lumen of the blood vessels, and it's surrounded by layers of tissue. We talked about the central lumen being this inside hole. We talked about connective tissues containing elastic fibers, and in the middle containing smooth muscles with more elastic fibers. We talked about arteries having more of these muscly fibers than veins. The elastic walls of arteries stretching and recoiling to accommodate the surge of blood after the contraction of the heart. We talked about veins preventing the backflow of blood, and when we talk about things like deep vein thromboses later, we'll look at an example of this. Hopefully that makes sense. It's just this little bit here that we're learning today. Please have a go at the Google Forms and ask any questions if you're stuck.